When you think about winding down at night, maybe taking one of those sleep gummies to quiet your racing thoughts, you're relying on a very specific chemical mechanism.
Yeah, exactly. Your brain uses a neurotransmitter, GABA, to just hit the brakes on your nervous system.
Right. It forces those hyperactive circuits to essentially just go to sleep.
It's a beautifully elegant off switch, really. GABA floods the synaptic clefts, hyperpolarizes the neurons, and suddenly, the biological volume dials down so you can rest.
But what if a tumor could, weaponize that exact same off switch?
That is a terrifying question, yeah.
I mean, we're talking about a cancer cell fundamentally rewriting its own biology to manufacture that brain sedative, pumping it out to literally put your immune system to sleep so it just can't fight back.
It sounds completely like science fiction, but it represents one of the most astonishing biological hijacking mechanisms we've seen in recent oncology.
Which is exactly why we are here today. We're unpacking some wild new research from Hernández-Verdin and a massive team of collaborators. It was published in a 2026 issue of Cancer Cell.
And it's a phenomenal paper. It looks at how GABA promotes resistance to immunotherapy, specifically in patients with TLS-positive tumors.
Right. So our mission for this deep dive is to dig into this bizarre phenomenon they call neuromimicry. We want to figure out why some of our absolute best, most cutting-edge cancer therapies are mysteriously failing in patients who, by all accounts, should be completely cured.
This research really does more than just solve a clinical mystery about immunotherapy resistance. It forces us to completely rethink the boundary between neurology and immunology.
Because the tumor is acting as a bridge.
Exactly. We are seeing a tumor act as a bridge between two systems we usually treat as entirely separate disciplines.
Okay, let's get into mechanics of this. Because to understand how the tumor fights back, we need to look at the immune system's heavy artillery first. Immune checkpoint inhibitors, or ICIs.
Right, the anti-PD-1 therapies.
Yeah. And if you follow oncology, these drugs essentially rip the blindfolds off our immune cells so they can recognize and destroy cancer.
But the holy grail with ICIs has always been patient selection. I mean, we need reliable biomarkers to predict who will actually respond to these incredibly powerful drugs.
Because they don't work for everyone, unfortunately.
Right. And that search led to the intense focus on tertiary lymphoid structures, or TLS.
I visualize TLS as these highly organized rebel base camps.
I like that analogy.
Thanks. Yeah, instead of fighting from the outside, the immune system sneaks directly into enemy territory, right inside the tumor's microenvironment, and builds a forward operating base.
And they're highly structured. You've got specific zones where T cells are primed and B cells are trained, activated, and supplied to wage war on the cancer cells right next door.
So that spatial organization is what makes them so lethal to the tumor.
Exactly. Clinically speaking, if a pathologist is reviewing a biopsy and spots these mature TLS base camps, it is historically fantastic news.
Because the infrastructure is already there?
Right. The presence of TLS is one of our strongest correlates for a favorable response to immunotherapy. The immune system is just sitting there waiting for the ICI drug to give the green light.
Which brings us to the massive paradox driving this entire cancer cell paper.
The non-responders.
Yeah, you have a subset of patients with these beautiful, structurally sound TLS base camps inside their tumors. They get the standard of care immunotherapy and nothing.
The treatment completely fails.
The tumor progresses just as aggressively as if the immune system wasn't even there.
So to figure out the why behind that failure, the researchers utilized the BioNIC clinical trial cohort.
BioNIC, right.
Yeah, it's this well-documented group of patients with metastatic clear cell renal cell carcinoma, so ccRCC, which is a type of kidney cancer.
Okay.
The researchers specifically isolated the biopsies of patients who had TLS-positive tumors, but were entirely refractory to the immunotherapy.
Okay, let's unpack this for a second with a quick recap, just to make sure we're all tracking.
Good idea.
So 1, ICIs are supposed to unleash the immune system. 2, TLS are those immune base camps built right inside the tumors. And 3, some patients have the base camps, the troops are deployed, but they are still inexplicably losing the war.
That sums it up perfectly.
So what did they find when they looked closer at those losing tumors?
They took those non-responding tumors and ran deep RNA sequencing to see what transcriptional programs were driving the resistance.
And what were they expecting to see?
Well, the assumption was that they'd find classic immunosuppressive pathways upregulated, maybe TGF-beta signaling or severe T-cell exhaustion markers.
The usual suspects.
Right. But instead, the data lit up with a massive, blindingly bright signal for something completely out of left field: GABA.
Wait, I want to pause on this. GABA. Gamma-aminobutyric acid.
The very same.
We're talking about kidney cancer here. Why is there a massive spike in genetic signatures for GABA synthesis, transport, and receptors in a renal tumor?
It's a great question, and it's what shocked the researchers, too.
And practically speaking, if a tumor is pumping out massive amounts of a neurosedative, why isn't the patient just falling asleep? Is this systemic, or is the tumor keeping this chemical warfare strictly localized?
That's the critical distinction. It is intensely localized. The tumor microenvironment is essentially acting as a sealed chemical reactor.
Oh, wow. So it's not spilling out into the rest of the body.
No, the GABA isn't flooding the patient's bloodstream in quantities high enough to cross the blood-brain barrier and cause systemic sedation. It's being concentrated directly in the interstitial space of the tumor.
Wow.
And the clinical correlation was undeniable. Patients with these high GABA signatures had drastically shorter progression-free survival compared to those with low GABA signatures, despite both groups having TLS base counts.
Okay, so if you are listening to this and connecting the dots, think about it. If GABA is the ultimate biological sedative and it's heavily concentrated right where the immune system is trying to work, what happens to those immune cells? What stands out to you so far?
It leads to an absolute subversion of the immune response.
Right. The tumor is bypassing standard physical blockades and opting for chemical warfare borrowed directly from the central nervous system.
Which immediately sets up a whodunit scenario.
Exactly. We know there's a localized GABA cloud putting the immune system to sleep, but who is manufacturing the sleeping pills?
Tumors are heavily vascularized and highly innervated. So if you're hunting for a neurotransmitter, the nerves infiltrating the tumor are the obvious first suspects, right?
Yeah, that makes sense. Neurons naturally produce GABA.
That was the immediate hypothesis. So the researchers analyzed the genetic signatures of Schwann cells and sensory neurons within these kidney tumors.
I imagine it's tough to isolate that signal in bulk tissue, though, isn't it?
It is, yeah. But even when they drilled down, it was a complete dead end. There was absolutely zero correlation between nerve density and the high GABA signatures.
Seriously?
Yep. Nor did nerve density correlate with the failure of the immunotherapy. The peripheral nerves simply weren't responsible for the massive GABA production.
So they had to look closer. And the technology they used to do this is genuinely staggering. Visium spatial transcriptomics.
Oh, it's an incredible tool.
For anyone who hasn't worked with this tech, usually when you sequence a tumor, you're grinding it up into a biological smoothie.
Right, bulk sequencing.
Yeah. You can see all the ingredients, the RNA transcripts,
Yeah.
but you lose all the context of how those cells were arranged.
Yeah.
But Visium spatial transcriptomics is like overlaying a Google map onto the tissue slice.
That's a perfect way to describe it.
You get the high-definition molecular data, but you can see exactly which specific cell is making which chemical mapped out to its precise GPS coordinates on the slide.
And the spatial resolution of that technology is what cracked the case. By mapping the tissue, they realized the true culprit wasn't an infiltrating nerve cell at all.
Don't tell me.
It was the cancer cells themselves.
Like the ultimate betrayal.
It really is. They identified a very specific cluster of malignant cells that structurally maintain some features of normal kidney tubes. They termed them proximal tubule-like or PT-like tumor cells.
Okay, PT-like tumor cells.
And the spatial map showed these specific cancer cells were actively and aggressively expressing an enzyme called GAD67.
Oh, wow. GAD67 is the smoking gun here. Glutamate decarboxylase 67.
Exactly.
If we look at fundamental biochemistry, its sole purpose is to take a very common amino acid glutamate snap off a carboxyl group and convert it directly into GABA.
And this brings us to the core concept of the paper. Neuromimicry.
Neuromimicry, I love that term.
What's fascinating here is that the tumor cells are undergoing a profound phenotypic shift. They aren't just mutating randomly, they are actively mimicking the highly conserved metabolic pathways of inhibitory neurons in the brain.
Just to carve out a localized zone of immune suppression in the kidney. If you're a pathologist looking at this Visium spatial map, it has to be a mind-bending moment. You're looking at malignant kidney cells, but they are suddenly running the metabolic software of a brain cell.
It really blurs the lines of what a cancer cell is capable of.
All right, let's do another quick recap, bullet summary for you listening at home. 1, nerves aren't the source of the GABA. 2, these PT-like kidney cancer cells are synthesizing the GABA themselves using the GAD67 enzyme.
Exactly. They are running a counterfeit pharmacy.
So the obvious question now for you, the listener, is if the tumor is manufacturing this chill pill, Who exactly is it trying to sedate?
To find the victims, the researchers used that same spatial map to look at the TLS base camps.
Right, back to the rebel bases.
Because they had the GPS coordinates, they could physically measure the distance between where the GABA was being produced and where the immune cells were stationed.
They mapped out what we could call spatial danger zones.
Exactly, yeah. They categorized the TLS into two distinct spatial groups. They looked at base camps that were physically distant from the GABA-producing tumor cells and compared them to those right next door.
The ones sitting directly in the blast radius of this chemical weapon.
Right. They labeled those PT-like close TLS, and the functional difference was night and day.
How bad was it?
Well, a healthy, fully functional TLS should be aggressively maturing B cells into plasma cells. Those plasma cells are the factories that pump out high-affinity IgG antibodies to coat the tumor and mark it for destruction.
Right, that's their job.
But the PT-like close TLS, they were structurally stunted, they were highly immature, featured drastically fewer plasma cells, and profoundly lacked those vital IgG antibodies.
So the sheer proximity to the GABA is stunting their development.
Yes, but seeing that on a tissue slide is just circumstantial. The researchers had to prove the direct biological causality.
So how did they prove it?
This is where they moved to an in vitro model. They took healthy human B cells, put them in a petri dish, artificially activated them to mimic the TLS environment, and then just bathed them in GABA.
Just soak them in it.
Yeah. And the resulting data was a bloodbath for the B cells. But the brilliance of Hernández-Verdin's team is that they didn't stop at observing the damage. They meticulously mapped the molecular sabotage.
Okay, I want to really dig into this mechanism. It unfolds in two distinct phases, right?
Yes, a two-pronged mechanism. Let's start with phase 1, the metabolic shift. This happens relatively early, around day 4 in the assay.
Okay, what's happening on day 4?
We have to look at the TCA cycle, the Krebs cycle. It's the central metabolic hub of the cell, generating both ATP for energy and critical biosynthetic precursors.
Right, standard biology.
When a B cell activates, it relies heavily on the TCA cycle to power its expansion and to present antigens using a surface molecule called HLA-DR.
So HLA-DR is like the radar dish they use to show the other cells what to attack.
Exactly. But when GABA enters the B cell, it fundamentally alters the metabolic flex. It forces a massive shift, specifically funneling alpha-ketoglutarate away from its normal cycle and converting it into glutamate.
Okay, wait. Here's where it gets really interesting. It's less like running out of gas and more like the tumor is maliciously rewiring the B cell's internal assembly line.
That's a great way to put it.
By forcing the cell to hoard glutamate, it starves the specific pathways needed to build and transport that HLA-DR molecule to the surface.
Yes. And if we think about immune function, without HLA-DR on its surface, the B cell becomes functionally blind and mute.
It can't hold up the tumor fragments to show the T cells what they need to attack.
Exactly. The metabolic stalling isolates the B cell from the rest of the immune network.
Wow.
But that's only the early phase. The late phase is receptor-driven.
Okay, so what happens next?
By day 7 and day 10, healthy B cells should be proliferating wildly and secreting massive amounts of antibodies. Instead, the external GABA begins binding directly to specific receptors on the B cell's membrane.
Which receptors?
The GABA receptors.
Now, wait a second. Why would a B cell, an immune cell, even express a GABA receptor? I mean, that seems like a massive evolutionary design flaw.
It seems like one, yeah. But it speaks to the deep interconnectedness of the neuroimmune axis. Immune cells possess many neurotransmitter receptors because the nervous and immune systems have to crosstalk during systemic inflammation or stress.
Ah, okay, so the tumor is just exploiting a back door that evolution left open.
Precisely. And the researchers proved this late-phase mechanism with a brilliant control experiment. They introduced a pharmacological drug called saclofen into the petri dish.
Saclofen.
Yeah, saclofen is a competitive antagonist. It physically blocks the GABA receptor.
So it plugs the keyhole.
Right. And the moment they added saclofen, nullifying the GABA signal, the B cells woke up and started proliferating again.
Unbelievable.
The GABA receptor functions as a hardwired sleep button. When GABA binds to it, it arrests the cell cycle, stopping division, and completely blocks the B cell's ability to secrete both IgM and IgG antibodies.
So to weave this complex mechanism together, let's do a quick mini recap.
Let's do it.
Number 1, proximity to GABA ruins TLS maturity. Number 2, the tumor pumps out GABA gas. The B cells in the rebel base camps breathe it in. Phase 1, their internal TCA assembly line is rewired. That alpha-ketoglutarate to glutamate shift strips them of their HLA-DR radar.
Right.
And then phase 2, the GABA hits the GABA receptors on the outside of the cell, hitting the sleep button and halting antibody production entirely.
It's a beautifully sinister two-step paralysis. You starve the metabolic engine, then you throw the biological off switch.
It's just wild. Let's pull back for a second, though, and look at the strategic vulnerability here.
Okay.
If you're a listener tracking the tumor's logic, you have to ask yourself, if a tumor relies on this one specific, intricate chemical trick to survive an immune attack, doesn't that also expose a massive weak point?
It absolutely does. If we know the weapon, we can disarm it. That is the ultimate translational hope of this paper. The researchers have mapped the evasion tactic. The next logical step is to see if we can drug it.
But before we cure it, we need to know the scale of the problem. I mean, is this GABA neuromimicry just a weird quirk of clear cell renal cell carcinoma? Or is this a universal evasion tactic used by other cancers?
To answer that, the team looked far beyond kidney cancer. They analyzed genomic and spatial data from the Pembrozarc trial.
Pembrozarc, so sarcomas.
Yes. This trial focused on soft tissue sarcomas, cancers originating in muscle, fat, and connective tissue. These patients were treated with pembrolizumab, which is another standard anti-PD-1 ICI.
So a completely different tissue origin, different tumor microenvironment. What did they find?
The exact same molecular signature. The non-responders in the sarcoma trial exhibited the identical GABA resistance pathways.
Wow, that's huge.
It is. The neuromimicry seems to be a conserved evolutionary strategy across wildly different tumor types.
That is massive validation. I mean, solving this doesn't just help kidney cancer patients. It opens up a new paradigm for immunotherapy resistance across the board.
Exactly. So they moved to test a cure using an incredibly robust in vivo animal model, the MCA-OVA mouse model.
The MCA-OVA mouse model. I love how enthusiastic you get about mouse models.
Well, it's a great model. It's a highly validated mouse sarcoma that naturally produces GABA and forms those crucial TLS base camps.
Perfect for testing this.
Right. To intervene, the researchers utilized a compound called 3-MPA, or 3-mercaptopropionic acid.
Okay.
The crucial thing to understand about 3-MPA is that it is not a cytotoxic chemotherapy. It doesn't kill cancer cells directly. Its sole mechanism of action is selectively inhibiting the GAD67 enzyme.
Oh, so it shuts down the tumor's counterfeit GABA manufacturing plant.
Precisely. It stops the synthesis at the source.
That's brilliant.
Now, you might think shutting down the GABA factory would be enough to cure the mouse, but when they administered 3-MPA as a monotherapy, the tumors kept growing.
Wait, really? Why?
Because removing the GABA doesn't kill the tumor, it just wakes up the immune system. The magic happened when they combined the 3-MPA with the anti-PD-1 immunotherapy.
Ah, so how good were the results?
The synergy was staggering. The combination therapy reduced tumor growth by 75% compared to the control group.
75%. That is incredible.
And when you look at the microscopic changes driving that tumor shrinkage, it validates the entire paper.
Yeah.
Inside the TLS base camps of the treated mice, the B cell density skyrocketed.
So the cells rushed back in.
Yes, the ratio of immune cells completely inverted in favor of B cells, and those cells were finally maturing rapidly into the antibody-producing plasma cells they were always meant to be.
The rebel base was back online and fully operational.
Exactly. And just to be absolutely scientifically rigorous, they proved this massive therapeutic rescue was entirely dependent on the B cells.
How'd they do that?
They ran a parallel experiment where they gave the mice a specific antibody that depleted all their B cells and then administered the combo therapy. Without the B cells present, the 3-MPA and immunotherapy combo lost almost all of its effectiveness.
It's the definitive proof of concept.
Yes. The ICI therapy is necessary to take the overarching brakes off the immune system, but the 3-MPA is absolutely required to wake the localized B cells up from their GABA-induced coma so they can actually execute the attack.
So what does this all mean? Let's do our final mini recap before we close out.
Sounds good.
Number 1, GABA resistance happens in sarcomas too, not just kidney cancer. Number 2, blocking GABA synthesis with 3-MPA restores B cell function. And 3, combining GABA blockers with ICIs shrinks tumors dramatically in mice.
Spot on.
So if we pull all these threads together, we connect this to the bigger picture. What's the ultimate takeaway here?
The most profound conclusion here shifts how we view cancer heterogeneity. For decades, oncology treated tumors as a relatively homogeneous mass of rapidly dividing mutated cells.
Just a lump of bad tissue.
Right. But this research proves that the tumor microenvironment is an incredibly complex, adaptive ecosystem. By uncovering how a tumor can weaponize a simple neurotransmitter to create a localized zone of immunosuppression, we can revolutionize patient screening.
So we can look at a patient beforehand and know what to do.
Exactly. If a patient presents with a TLS-positive tumor but high GABA signatures, we now know that standard immunotherapy will fail. We need to implement a rationally designed combination therapy that blocks the GABA synthesis first, clearing the chemical fog before we send the immune system in.
It's the absolute pinnacle of precision medicine. We aren't just carpet bombing the body anymore. We are countering the tumor's specific molecular hacking.
Exactly.
And for you listening, whether you are a clinician trying to parse out why a patient isn't responding, or a researcher mapping these dizzying cellular pathways, or just someone fascinated by the sheer ingenuity of biology, this deep dive is a stark reminder of how deeply interconnected the body truly is.
It really is one giant system.
Yeah. The rigid lines we draw in medical textbooks between, neurology and immunology, those are human constructs. They don't actually exist in nature. The body is one giant, highly communicative network, and cancer is the ultimate opportunistic hacker of that network.
It survives by taking the body's most elegant regulatory codes and turning them against the host.
Which leaves me with a final, slightly unsettling thought for you to mull over. We've just spent the last 20 minutes exploring how cancer cells can dynamically evolve to hijack our brain's calming neurotransmitters merely to put our immune system to sleep.
Right.
So think about the sheer scale of the body's signaling networks. What other everyday biological signals, what other obscure hormones, peptides, or brain chemicals are tumors secretly manipulating right now in ways we haven't even thought to look for yet?
We are just barely scratching the surface of the tumor microenvironment.
Keep that in mind the next time you pop a sleep gummy and wait for the GABA to kick in. Thanks for joining us on this deep dive.